Alrighty, so this is Unit 1. It's the basics and it's chemistry, so it shouldn't be too terrible, and you should remember most of it, and we've used a lot of it as we've gone through the year. And so the first stuff is just water and hydrogen bonds. And so we're going to talk about biological molecules. There's 4 of them. Each one, well, not each one, but for the most part, they have monomers and polymers. The monomer is just the one, and poly is the many, just what the prefixes mean. This little chart is good as sort of an overview of the 4. But if you see under lipids, there's no monomers or polymers. It's just lipids are a little bit different. And so, but if you see like the monosaccharides, polysaccharides, and then their energy, nucleic acids are genetics, and amino acids are protein, and then polypeptides, mostly proteins, primarily in our bodies, structural, and then enzymes. Alrighty, so this slide talks about polarity. That's when one portion of the molecule has a slightly positive charge and the other has a slightly negative charge. So if you look at the little figure, the oxygen is slightly negative and the 2 little hydrogens are slightly positive. So that gives us imbalance in sort of the pull of the electrons, and then the little regions become slightly charged. And so then they attract or repel accordingly. Looking at the little GIF, when water freezes, it becomes less dense than the liquid form because of the polarity and how the negative portions are attracted to the negative portions. And so then it makes like a little lattice crystalline structure, and then the little molecules spread out or are forced to be spread out. So then it floats. And so hydrogen bonds technically aren't a bond. It's just a weak attraction between the hydrogen on one and the something on another. And so that's why it's called a hydrogen bond. And then the hydrogen bonds are between the hydrogen of one molecule and the oxygen of a different molecule, not between the hydrogen and the oxygen within the same molecule. Here's a couple videos. I would click on the links. The top one, the launch pad, just shows the difference between adhesion and cohesion in a cool little video on the space station. And then the penny in the water activity we did in class. It's when we took water, plain water with soap in it, and then see how many drops we could put on top of the penny before it spilled over the edges, and we took a little bit of data, and then we talked about how the cohesion and adhesion was affected. The next one is the elements of life. And so there are 4 big ones that we're gonna, or 5 big ones that we're gonna talk about. My head is in the way of phosphorus. But it is carbon, nitrogen, oxygen, hydrogen, and phosphorus. The 4 macromolecules that we're gonna talk about contain these, some all of them, some 3 of them, but we'll just talk as we go through. So the first one is carbon. We are carbon-based. Carbon is in everything that's in a living thing. When we talk about organic chemistry, that is the chemistry of carbon-containing molecules. Carbon is useful because it has 4 valence electrons, so it can bond with a lot of different things in a lot of different ways. So it is flexible and useful. So we have evolved to be based in carbon. Nitrogen has 5, and phosphorus both have 5 valence electrons. It just they're needed for different things. We'll talk about those in a bit. But like ATP contains phosphorus because it has 3 phosphates. The nitrogen is in nitrogenous bases, which is in nucleic acids. So we'll just look at those a bit as we go. And then to talk about the monomers and the polymers, and then changes. So organic molecules, we talked about this just a second ago. Carbon is very useful because it can bond with 4 different things, or 3 or 2 or 1. It can form different kinds of bonds. It can form rings and chains and all kinds of different things. So we can make a whole lot of stuff out of it. And then there are 4 things for biomolecules or organic molecules or biological macromolecules, however you look it up, it all means the same thing. And then monomers are the one part and the polymers are the many parts stuck together, so we'll talk about the different examples in a second. There are two kinds of equations that we need to remember: condensation and hydrolysis. Condensation is when we are making water. So when, like if we have a cup, I'll use my cup as an example in this. So if this were to get condensation or like water on the outside, we would be making water is how I sort of remember that. And so a product of a condensation reaction, one of them is going to be water. And then hydrolysis is the opposite. We are using water and incorporating water to split something. So we're going to start with one thing, add some water, and then end up with two things. And so I'll show you a picture. And so on this one, this is condensation because we are making water. We have two things that are coming together and we're getting rid of the water. And so I think of it as making water. And then the opposite is hydrolysis. So we use the water to lyse. And so we have one thing and turns into two things. And then whenever you break those bonds that were bonding them in the top molecule, then there's sort of open spots for bonding. And so then we have to fill in those open spots with our oxygens and our hydrogens. So the four different ones, the first one we're going to talk about is carbohydrates. Carbohydrates are, there's the most things out there, plants, structures of pieces of like bugs, and then lots of stuff in our bodies is carbs. We use carbs primarily as the energy source. Glucose is the big one. In the next couple of units, we'll talk about cellular respiration. So we'll talk about glucose more, but carbs are a 1 to 2 to 1 ratio. So it's like glucose is a C6H12O6 ratio so you can, it's molecularly, it's a little bit easier to identify those. If I were to give you structurally or molecularly a formula, you could say, oh, that's a 1 to 2 to 1. So then that would be a carb. And then just some examples with some vocabulary at the bottom, just so to be familiar with. And then monosaccharides is one. It's small, it's simple. This would be glucose. There is, it's the smallest unit. There's more monosaccharides than just glucose, but glucose is the big one that we talk about. Oligosaccharides are short chains. Sucrose is glucose and fructose stuck together. And so then we get a disaccharide, pretty easy to understand. It's just two, but it's just short. And then polys are many, so that's a bunch. And so there's a few listed on here, and then I'll show you a few slides that talk about those individually. And so cellulose is plant materials. It's mostly structural. It is like the stringy stuff in celery. That's cellulose. So you like chew it, and it doesn't chew real easy. It doesn't dissolve real easy. It's also the outside of like corn and the outside of peas and some of the different parts of peanuts, but doesn't dissolve. So it's usually rigid and usually structural. Starches is energy reserve. So it's a polysaccharide. This is like potatoes or different starches like that. Glycogen is a special kind of polysaccharide that we store in our muscles and our liver, so that if we don't have enough glucose, we have a little backup. And so it's a short-term sort of reserve that we don't use on a regular basis, but it's there evolutionarily sort of to save our lives so we don't die. And then chitin is a rigid structural material, and then so think of like a crab on the outside, and then it also helps fungi. And then lipids are the next biomolecule that's fats and oils and you know what lipids are, waxy stuff. It's usually a protective coating, excuse me, or something that gives sort of fluidity, but we'll talk about those when we get to membranes in the next unit or so. Two kinds that I want you to remember. So when we add them up and we make a what you might think of like a polymer, but there aren't really polymers for lipids. But when we add a bunch of them together, we can get saturated or unsaturated fats. Saturated fats means that every carbon, every spot has as many hydrogens as it can hold. That it's saturated with hydrogens. Unsaturated is when there's a double bond, and so then we have not as many as could possibly be attached for the carbons or for the hydrogens. And then what that results in is structurally they're different, and then sort of how they behave is different. So saturated fats are solid at room temperature, and unsaturated are liquid at room temperature. So just a couple of pictures. And so the one on here, so the one A would be a saturated, the B and the C would be unsaturated. And so just some examples of more stuff about fats is there are, it has tails that it has attached to it. And so a triglyceride has 3 tails that is attached. So there's a picture, but they're all hooked together. Alrighty, so phospholipids are in cell membranes. The head part, the sort of top circly part, is hydrophilic, means it likes water. Hydrophobic tails are the ones that don't. And so when we are in a water environment and we make a phospholipid bilayer, the heads go towards the outside and then the tails fling in, and then we get a bilayer. And so just a picture. And then wax is another kind of lipid, and then it's, you know what wax is, but just gives you a little picture of that little bee and a honeycomb. And then, but like the outside of like thick aloe leaves or stuff like that has like a waxy outer covering. And then steroids are a one of the biological lipids that we care about. And so we have steroids like estrogen and testosterone, but cholesterol is also one of them. Cholesterol gives us fluidity in a cell membrane. And so if we have all of our phospholipids all sort of lined up, we can shove a cholesterol in there and it sort of pushes these apart. And so it gives them the ability to flow whenever, so then it makes it so that our cell membranes aren't too rigid. And then just a picture of molecularly those two. And then you know that there are external traits that we get from hormones. So this is just an example in ducks. In humans, it would be like hair on your face or not hair on your face, or sort of a bigger muscle mass or not a bigger muscle mass. Just, you know, certain sex characteristics, those come from estrogen and testosterone. And then proteins and proteins is the next macromolecule. They are made up of amino acids. And so we didn't talk about this a bunch, and AP doesn't really ask about it a whole lot, is the sort of nitty-gritty of the R groups. But we have an amino group, and we have a carboxyl group, and then we have an R group. And the different R groups give us different amino acids. And then we have just a picture to go with that. Oops. There you go. And then polypeptides are just proteins. They're held together with peptide bonds. And then if we look on here, it says amino acids, so the single pieces are bonded together by peptide bonds in a condensation reaction between an amine group and a carboxyl group of another, which means that when we make proteins, we also get water. And so then there are 4 levels of structure for proteins, and you sort of know it, but know how to apply it, and sort of when we mess with things, how it affects these structures. So we had an FRQ on this, so you might want to take a look at that, or we'll talk about that later. So the primary structure is just the amino acid sequence. The secondary structure is when it folds and forms little bonds, and it sort of gets a little bit more organized. Tertiary is when it binds up and folds up and squishes to give us a better, or a structure that works. And then the fourth one is when we have more than one of those tertiaries, like a tertiary of this, tertiary of this, and then they come together and then they make a quaternary structure. So it would be more than one protein stuck together. And then it just gives you another example of a big, huge something, so hair. And then whenever we mess with proteins, specifically what we care about is enzymes. There's four big things that do this. We messed a little bit with heat, and I think a little bit with pH in class, but the other two do the same thing. So heat changes in pH if it's too acidic or too basic. Salty environments or detergents can mess up the hydrogen bonds, and so if you can't hold together, then you can't maintain a shape, and so then it becomes denatured, and if the shape isn't right, then the function isn't right, so then it doesn't work anymore. And then for nucleotides is another macromolecule. So nucleotides make up nucleic acids, and so we have a sugar, a phosphate, and a nitrogenous base. Weirdly, but maybe not so weirdly molecularly, ATP falls under the heading of nucleotides, and so a nucleotide with 3 phosphate groups, and so structurally, molecularly, it's the same thing. And then, of course, we know ATP is for energy, but we'll talk about that with cellular respiration. All right, so then this is just a picture of ATP. We have sugar, we have a phosphate, and we have a nucleotide. So if you sort of look at that, but then we have little extra phosphates over there. So, but the other is very, very similar to a nucleotide, like we think of when we think about DNA. And then nucleic acids are just the polymers of those. So we add up a bunch of A's, T's, G's, and C's with our sugar phosphate backbone when we get an acid. So it would be the polymer. And then RNA is one of them, and so it's usually single-stranded. It has uracil instead of thymine. It's used in protein synthesis because it's how we make messenger RNA and get it out to the ribosome, all that stuff. And then DNA is a double helix. It's heredity. It's in the nucleus. It's stuff that you know, but instead of U's, we have T's.